In this experiment, we're going to test the effectiveness of various antibiotics, and we're going to compare them between a gram-positive and a gram-negative bacterium. This test is sometimes called a susceptibility test, also known as a Kirby-Bayer test. And what you're going to do is you will take a petri dish that has antibiotic agar on it, is kind of the nickname that we use for it. And you are going to inoculate that plate with the bacteria that you're testing. You want to use a cotton-tipped applicator and create a bacterial lawn. Remember, this is where you dip the cotton tip into the broth containing the bacteria, and then you want to spread that completely over the surface of that petri dish. You want it completely covered so that when it would grow, you create that lawn, that complete covering of bacteria. Dispose of the cotton-tipped applicator properly, and then what you will do is you will take your antibiotic discs. You decide which antibiotics you're going to test. These discs are similar to the paper discs that we use when testing chemical agents. They're already pre-soaked with the antibiotic in them, and so you just place them using aseptic technique on top of the surface, just lightly tap it so it sticks to the surface of the agar. You want them about equidistant from each other. You can usually fit about 5 of the disc on the plate, and then you will incubate the plate. After the incubation period, you will then look at the plates and measure the zone of inhibition. Remember, the zone of inhibition is that area of clearing where there is no growth surrounding a disc. If growth is all the way up right to the disc and there's no zone at all of inhibition, then that means that that antibiotic is not effective at all against that particular bacteria. If there is a zone of inhibition, it's inhibiting the growth, which means that antibiotic is preventing the growth of the bacteria, and so it's effective. The larger the zone of inhibition, the more effective that antibiotic is. And we do see differences between gram-positive and gram-negative bacteria and how they react to antibiotics. So when you go to look at your results, you have worked with 1 gram-positive plus 1 gram-negative bacteria. You would have tested the same antibiotics so that you can compare the effectiveness of them. Just so you know, most antibiotics that we have are effective against bacteria. There are a few antibiotics that are effective against fungi, but it's harder to develop antibiotics that are effective against fungi. And the reason for that is because fungi are eukaryotes. So are our cells. So the fungal cells are similar to us. The ribosomes are the same size. We both have mitochondria. It's harder to find an antibiotic that would be effective at killing fungi, or at least stunting their growth some, but doesn't do the same thing to our cells. So obviously you want to kill the fungus without damaging our cells, and that's a little more difficult to do. Also, there are a few, but not as many antibiotics against viruses, and the reason for that is viruses typically are intracellular. They spend most of their time inside the host cell. So how do you even know if a cell has a virus in it? And even if you detect, oh, this particular cell has a virus in it, how do you kill the virus without killing the host cell or even damaging the host cell? That also becomes very hard and tricky to do. So for that reason, most of the antibiotics that we have are effective against bacteria. Sometimes when we talk about antibiotics, we talk about narrow range or narrow spectrum and broad spectrum. And that just is referring in general to how effective are they. A broad spectrum is effective against a wide range of organisms versus a narrow spectrum, as the name implies, is only effective against a few. In this slide, these pictures are showing over here you have this larger plate right here is a gram-positive, and over here on the left it's a gram-negative organism. You have the same antibiotic disc on one plate as on the other plates. You notice that the gram-negative, a couple of these have a very narrow zone of inhibition, but the antibiotics are definitely much more effective against the gram-positive organism. And this right here has the largest zone of inhibition, so this would be, of these choices, would be your best choice to use, the most effective in controlling the growth of this particular bacterium. And on this slide, we're showing once again how you have this nice large zone of inhibition, and you actually measure from the disc out to the edge to get a number of what the distance of that zone of inhibition is, so you can compare the different antibiotics from one to another, can compare between the organisms.